My first 9 grants were rejected, and I had trouble getting faculty positions. I think that we're in the investment community didn't think it was possible. So we're looking at mRNA in many other areas. The biggest ones I'd say are cancer, ways of doing new kinds of cancer therapies. That if you're not your own champion, no one else is.
We have invited Dr. Robert Langer. He's one of the 9 MIT Institute professor, which is the highest faculty rank within the school academic system. He's also one of the few people who is the winner for the U.S. National Medal of Sciences in medicine, engineering, and also science for Queen Elizabeth Prize of Engineering and Breakthrough Prize in Life Sciences. And on top of this, he has over 220 other awards. He's both adored by the White House and also in the UK, the royal family. So today we really get to have a behind the curtain conversation with Dr. Langer to ask about his journey and how he can inspire the next generation of scientists. One of the journalists have written, I think about a couple of years ago, that Dr. Langer owns up to 1,400 and more patents and have licenses in more than 400 companies. The impact he makes in patient life cumulatively in the chronic disease space can really measure up to 2 billion patient lives. And that is a data that was reported by a journalist about 7 years ago. And I can imagine, now post-COVID, with his establishment as the co-founder for Moderna, the number must double or even triple. So we are very honored today, Dr. Langer, to have you here to share your incredible journey of how you shape the industry. And we really wanted to kind of be able to kind of dig into your mind and to think about what can we learn from your journey as young scientists and entrepreneurs.
Well, thank you. It's a pleasure to be here and happy to try to answer any questions.
Sure. So we wanted to kind of start off. I've been following your podcast, a lot of podcasts. I interviewed you and your journey for quite a bit, but we have a mixed audience here. We have people who are scientists, people who are from the business school on the entrepreneur side, and also people from industry. So we can start off really trying to understand you previously in your early years with the postdoc in Bachman's laboratory, and you were studying at intersection of medicine and surgery. And you had a breakthrough in that discovery, really leading into a new field of biochemistry. So could you share a bit about your early journey from scientist to entrepreneur? How did you transition? How does your approach to innovation change over the years?
Yeah, well, of course, when I first started, when I was probably a lot of your ages, I really wasn't even sure what I wanted to do. I ended up getting this job, as you mentioned, at Boston Children's Hospital. And we were trying to actually, Dr. Folkman had this idea that tumors made a substance that induced blood vessels to grow. And he had a further idea that maybe there'd be chemicals that could stop it. It was a very controversial idea. Almost everybody felt he was wrong. Of course, I didn't know any biology, and I thought it sounded great. But my job was to prove he was right, and in so doing, isolate the first blood vessel inhibitors. One of the challenges in doing that was to create a bioassay to study that. And without spending too much time, one of the keys to that was creating tiny little particles that could deliver large molecules, which is what we thought would be these, the blood vessel stimulators or inhibitors. People thought that was impossible to develop such a little delivery system. I was able, after hundreds of failures, to finally succeed, which we published in Nature in 1976. And around that time, we also published in the journal Science in 1976 that there were blood vessel inhibitors based on using, those little systems to deliver the molecules we found. I kept doing research. In fact, people didn't believe that the things I did were right. My first 9 grants were rejected, and I had trouble getting faculty positions. But eventually, over the years, those things happened, and I just kept trying. I'd say maybe about less than 10 years after we started, we actually filed for a patent at Boston Children's Hospital, which at that time had no patents. And that patent got rejected about 5 years in a row. And we kept trying, and I actually eventually found a quote in the literature that said what we were doing was extremely surprising. And we showed that to the patent examiner, and he allowed the patent. So then that took about another 6-7 years before we got the patent. But around after we did, people started to call me up from large companies and want to use these kinds of approaches for things, which I was very excited about. And we got grants, and I consulted, and most exciting to me was those companies were going to develop it. But what happened is, they worked for a year or two on it. They didn't get exactly the results they wanted, and they just gave up. And so one of my friends, Alex Klebanov, said to me one day, well, Bob, we should start our own company. So we did, with several of my students and with Alex. And, that company did okay, but we kept doing it. I could see from these experiences, now over 40 years ago, that if you're not your own champion, no one else is. And almost always I'd say that. And so that really is how I moved maybe from being a scientist to an entrepreneur.
Thank you, Dr. Langer. And back then in the mid-70s, you were working at a rare interface of medicine and chemistry, and you're probably one of the first person to really be able to delve into that field, which now sort of grew into the biochemistry larger field where me and most of my classmates are have a background in. So back in the day, what are the challenges of tapping into a brand new arch of medicine? Did you feel like, what kept you going was the fire under your belly, that that your hypothesis was going to work, or was it because that you just had the scientific drive to kind of to explore your hypothesis as a PhD? What was one of the two reasons?
Well, I mean, a couple of things. One, I really thought Dr. Volkmann's vision was so powerful. If you could stop blood vessels, could you stop cancer? And I also felt that the drug delivery work we'd done was also very important. And I think that those things from a science stand kept me going. I also finally did get a position as a faculty member, assistant professor at MIT. I also really liked working with the students, and I think that kept me going. So I think it was those kinds of things, the importance, if we were going to be successful, of those two areas. And eventually, of course, they ended up both becoming hugely successful in terms of making an impact. The angiogenesis work, that's led to drugs, some of the most successful drugs in history, like Avastin and Eylea. And the drug delivery work, well, that's led to things like we've done at Moderna and Alnylam and many other companies, because if you don't protect some of these molecules from being destroyed, well, they'll never be useful therapeutically. So I think that it was all that helped me.
So now, that's with that mindset is inevitable for the success for Moderna to come to place. In the early 2019, Moderna was the first company to really, in competition with Pfizer, to release the first COVID-19 vaccine, first mRNA-based vaccine. So previously myself, I used to work at Sanofi Pasteur, which is the vaccine arm for Sanofi. And I know the dosing process for a traditional vaccine can take over 10 years development process. Whereas the mRNA development process is really game-changing. The staggers out the process of vaccine development really shorten that sort of drug to patient timeline to a few months, which is quite incredible to see in the scientific world. Now fast forward to 2019. How did the idea behind Moderna first take place? Were you involved from the beginning, and how does it feel like to kind of move from professor now to be on the board or to be running a company? What are the early moments that define this trajectory?
Yeah, well, by 2010, I'd started a number of companies. Some did reasonably well, others not as well. But by that time, I think, quite a few products had been developed based on, the work from our lab and those companies. Derek Rossi was a professor at Harvard. He came over to see me because Tim Springer told him he should see me. He had made a finding that he could take a regular cell converted into what's called an iPS cell with messenger RNA rather than DNA, or rather than viral vectors, which had been done before. And he had talked about, maybe we could start a company. He tried. A lot of people before this had tried to start companies in the United States, including the people that won the Nobel Prize in 2023. But people, I think, that were in the investment community didn't think it was possible, partly because of delivery, partly maybe because of other things. Um, I thought it was possible. I mean, and Derek had also tried. I think he went to Third Rock and they wouldn't fund him. But I certainly thought it was possible. And I mean, Derek had different ideas. He thought maybe you could have a company that could make kits. Maybe it could do tissue engineering or stem cells. Maybe it could do proteins. I thought certainly of all those, the protein one made the most sense. And so we also got Nubar Fayan involved, who was chairman of Flagship Ventures, and Ken Chen, who is a well-known cardiologist. So the four of us started the company. Um, Derek, myself, Nubar, and Ken. And, we probably spent the first 6 months planning, filing patents. And then Nubar said to me one day that, there's a fellow named Stefan Boncel, who he knew, and I also knew. He said, well, maybe we should try to get him to be CEO. And we did. And then, we went from there, hiring other great people, including some of our students. And so Moderna started, like I say, in 2010. We kept growing, raising money, moving things into clinical trials, getting deals with big companies. By the time 2019 came and COVID came, we were in a pretty good position as a small company, to develop that vaccine. And that's what we did.
So Moderna started actually in 2010, so it was well prepared for the opportunity when it came around for the COVID-19. In 2019, when it expanded, the business model sort of for the Moderna company has changed. How did it grow so quickly into its IPO in the in the 2019-2020 years?
Yeah, well, we did the IPO, I think, at the end of 2018. And so we had, a certain amount of money from that. And of course, we had deals with Merck, AstraZeneca, and companies like Vertex. So we had a lot of things going. But of course, COVID, we wanted to try that too. Stefan felt that was an important thing to do. And I think we all agreed with him. So we made that a priority. And we had built a manufacturing plant, a pretty significant one in Norwood, that we used. And of course, now it's quadrupled in size, and we're making other manufacturing plants. But, we made that the priority. And, and of course, I'm very glad we did. It saved many, many lives.
So Moderna is definitely a very successful example. I work with my classmates. Some of them are, early stage founding company, and the process of taking an idea from bench to bedside is actually a long process. You have to go through the valley of death of translation into clinical trial. Looking at the idea initially in the in vitro setting, right, in the laboratory, what are the process of really validating and determining whether your scientific idea will be also clinically sound and will have an impact for patient outcome? Is there ways that Dr. Langer that you sort of secretly have developed over the years that you can share with us?
Yeah, well, I think what you do is like what you said, you start out in vitro and then you do small animals. I always think, for the most part, and it may depend on the disease, that really the key is non-human primates, showing that in a non-human primate that your technology is safe and effective. I think if you can show that, and again, cancer is probably unfortunately not the right disease for the non-human primate, but many diseases are. And, say at Alnylam, which I was also on the founding scientific advisory board of, and Moderna, I think the big step forward when other people would be willing to put up big dollars to fund us was having safety and efficacy in non-human primates.
Yeah, before we go into the question regarding your lab's research today, I want to kind of just to stay a little bit on the topic of the sort of the developing a biotech startup, sort of anchoring in today's landscape, funding landscape, and ask a few questions that can be very practical for the audience. As we know, Moderna, the success of Moderna has really brought a lot of confidence into, venture capital investor who traditionally would shy away from these long return scientific assets. So that has really opened door for influx of investment between, I would say, probably 2019 to the early 2020, where the funding is starting to kind of trickle down. And in terms of today, the policy has really been sort of trying to kind of, I would say, not very supportive of scientific innovation compared to back in 2019. So under this type of environment, would you think there could be other sort of financial mechanism or ways of getting early stage startup, biotech startup funding? And what are those avenues, and how do you sort of reach out and, really start funding your idea?
Yeah. Well, I'd say two things. First, what I've watched over the last now close to 50 years is really there's ups and downs in getting financing. And you're right. But I would say actually the last 3 or 4 years, have been very tough for getting financing. But it's not non-existent. I mean, we just announced, I think Tuesday, a new company, Souffle Therapeutics, where we've raised $200 million in venture money and up to $3.5 billion in company money to create.
Congratulations.
Oh, thank you. But I mean, I'm just trying to say that it's certainly possible. I mean, sometimes it's harder than others, and but I think if you have good ideas, good people, investors will do that. I mean, again, you have to have a reasonable progress and reasonable people. We are able to do that, and others are able to do that too. But I certainly think it's possible. And to your question, there are other mechanisms. I mean, first of all, the biggest mechanism for small companies, of course, has been venture capital. But sometimes you find private investors who are interested in it, partly maybe because of the diseases you're going after, because they've had people who they love who maybe are ill or who've died. There's also foundations. In fact, our lab at MIT, we've had funding from the Gates Foundation, from the Juvenile Diabetes Foundation, from the Helmsley Foundation, and they've been extremely helpful. And even the Juvenile Diabetes Foundation, now they have a different name, T1D, but they even have an arm that helps fund companies. And I'm just, giving you examples. I mean, people, there's actually many different ones.
Mm-hmm. So these are brilliant examples. And I think exploring options outside academic is really, it's going to help young scientists to kind of grow into like an entrepreneur stage entrepreneur. As some of the VC money has sort of becoming more tight, there are definitely, other, avenues that Dr. Langer mentioned that we can look into as scientists and students. So on to the next question that I've actually also sort of listen to a few podcasts and sort of understand that your early life in high school, your relationship with your classmates and your teachers, and you had a very famous quote. I think it was you said that while in school you had to be good at answering questions, but when you're a scientist, you have to ask big questions and know what those big questions to ask. What are some of the big questions that you're in your laboratory is currently asking? What is at the forefront of your work in your lab's research today?
Yeah. So actually what I said, just to go back, is what I said is like when you're in grade school and high school and even college, you're judged by how well you give answers, actually, but to other people's questions. But in life, I think what's really important is the questions you ask. So anyhow, some of the questions we're asking are, can we make brains, in vitro brains, to study what's going on in the brain? We're going to have an article coming out by Alice Stanton and Lee Wei Tsai at MIT and myself and others on creating really the first human brain in vitro with all 6 brain cell types and a blood-brain barrier. And I hope that we'll be able to use that to ask big questions about how can we someday cure Alzheimer's, ALS, other brain diseases? Can we find new targets? Another big question we've asked, this was done with Phil Sharp, who's one of my colleagues, is a Nobel Prize winner, and we've had a wonderful postdoc, Vikas Chauhan, and is when people do gene editing, some of the time they get errors called indels. And Vikash actually figured out a way, to almost essentially stop that, to decrease it by about a factor of 60. Now it's just published in Nature. But we'd like to go all the way and make sure there are never any errors when you do things like that. And we're also doing a lot of work, big questions with the Gates Foundation, to try to take some of our discoveries that have made a big impact in the developed world. Could they do that in the developing world, in nutrition and vaccines and so forth?
It's amazing. We'll follow closely to your progress. Now I want to travel a little bit back in history, looking sort of like a telescope lens, looking at the current where we are in history with all the policy changes in the healthcare world that impacted student lives, that also impacted people who depend on grant for research. And just looking back in history, and I also want to bring it back home, I think it's important nowadays with a lot of challenges that not the medical school faces, but also the greater community in healthcare faces. Delve and talk about approaches to adversity and failures. I always believe, that's what really propels you to growth into the next stage. It's going to drive today's talk home. We invited Dr. Langer to our Harvard community, but he is not affiliated with Harvard. Dr. Langer's father actually has a degree from Harvard. So he's long ago very affiliated with Harvard University. And we've also learned that, Dr. Langer, you're a very humble person. You have a brilliant father, good family, great wife, 3 kids. You're very humble and very generous to share your insights with us. Back in the days, your father's era, he was, living his career in the Great Depression era, right? So he was facing a lot of similar challenges, definitely more serious challenges regarding sort of just getting out of school and, trying to navigate a job market, but also, a career path that is filled with a lot of uncertainty and adversity. And he's able to shine through it. And just, I thought to kind of lead back to that topic. If you can maybe think back in those times, did you feel like there was some similar events? Was there any ways that you felt like you and your father had dealt really well with adversity, in those difficult times? And what advice you could give to students today facing the similar challenges to really shine and stick through these times?
Well, it's a very, very good question. My father, as you pointed out, was in the Depression, and he would tell me stories that people who were actually very wealthy would jump off buildings and kill themselves because because they'd lost all their money. If you watch the movie Titanic, that the guy at the end who was such a not very nice guy, he did that. My father said that happened frequently. But then it wasn't just the Depression. My dad fought in World War II for 4 years.
Yeah.
And, he got wounded, but he was, came back. He was there for 4 years. He won medals for that. But he always told me he saw a lot of his colleagues and friends die in the war. And he always felt every day he lived past World War II was a gift. I think that has always stayed with me, that he had that attitude. So I try to tell myself that, boy, I mean, sometimes things might look bad, but it's not like we're not in World War II. We're not in the Depression. So you do the best you can and you muddle through. I mean, I can't, things have always in the United States, sometimes they go down, sometimes they go up. And I think you stay the course. And I still feel like with science, if you do good fundamentals, you'll end up doing well. And it's not just my dad. One of the great things that I've watched with the United States is the entrepreneurship about never giving up, no matter what happens. One of the stories I've often liked was the one of Milton Hershey, and he had a very different, maybe even simple dream, which was starting a candy store. And 8 times in a row, I think, he tried to start a candy store and he failed. But the 9th time, he didn't. He just wouldn't give up. That kind of attitude is just so important, about never giving up.
Thank you for sharing, Dr. Langer. I think perseverance and resilience in this hard time is a gift and it's something that we should all hold on to. I can see that this mentality have sort of propelled you through the time where you were sort of partnering into a new field of science. And in laboratory, have you had an example where you've had failures and how did you overcome those? Or, how did you overcome mentally and also processes that you've adopted get you out of the trenches?
Well, you keep trying. When we did the nanoparticles, I probably succeeded on my 201st try. That's the only way you can do it, I think. You just have to keep hanging in there. I wish there was some simple way, but most things that are worth doing, they're not simple.
Yeah, no, agree. Okay. Yeah. So now we're going to wrap up and just to scoop some advice from you. This will be a treat for the audience as well. So from your perspective, I'm sure that people in the audience looking to kind of build their own stuff would like to know, what are the three sort of most significant unmet medical need that you think future innovators should focus on? So give them a sort of a directions of the
Well,
market.
I'd break that down into two things. I mean, I certainly think if you're looking at diseases and problems, brain diseases are way up there, heart disease and cancer are way up there. But I think from a research standpoint, what's really important isn't just the diseases. It's a lot of the things that we've done, a lot of the things that other people have done that have led to new treatments are really core discoveries that from basic science, or in my case, basic engineering or chemistry, that might create platforms that could be used for anything. So you look at how Moderna did things, that in delivery. Well, that didn't come out because we were trying to do that. It came from things we did 50 years before and trying to do that something else. So I think basic research that can lead to new knowledge, that can lead to new inventions, I think that's probably the most important thing you can do. When you do that, you can apply that to a lot of those things to almost any disease. But if I were picking diseases, like I say, I'd pick brain diseases and I'd pick cancer and I'd pick heart disease.
Got it. Thank you so much, Dr. Langer. We have 10 minutes for Q&A at the end. And I'm going to pass it to Michael.
The first question is, Dr. Langer, you've always been a scientist at heart, yet you've successfully built and founded multiple biotech companies. How did you manage to raise money without a business background? Do you have any advice on biotech startups kind of failing during fundraising? What do you think made the biggest difference in helping your venture succeed where most don't?
Well, I mean, I've had failures too. Investors look at good science, and I've been lucky. When I started, investors thought the things that I was doing were worth investing in. It wasn't like that I was a good business person or even a good fundraiser, but the investors, I think, looked at people who they think are doing good science. I mean, really, that happened over the years. I hope I know a little bit more about business than I did, in the 1980s. But really, when I've done these companies, if to the extent we've succeeded, it's been a team effort, not just me. I like to think that I can help attract good scientists, but I've really worked with great business people, great investors, like Terry Maguire at Polaris, like Nubar Fayan at Flagship, like Mark Levin at Third Rock, and people like that. And then we've hired great CEOs like Stefan Bancel and Alan Crane and others. I think that it's really the team effort and that enables you to succeed.
Yep. And just a follow-up question. Since early stage research, you really don't generate profit right away. How do you manage the tension kind of between scientific exploration and investor expectations? And what helped you to keep both sides aligned in the early years?
Well, I think that investors in the medical area are interested in value. It doesn't necessarily mean sales. And I think that's been an important thing in biotech. If you make progress towards your goal, get good animal data, maybe even get good human data, and get good patents, maybe get good deals with partner companies, that's progress. And I think that if you can do that, investors will be happy. The other thing I think that's important to realize is the biotech industry is such that if you do a really good job, there's enormous value, even if you have no sales. There are companies that I've been involved in that have had no sales and have been acquired for $14 billion by a large company. The reason they are is that people expect there will be those sales. They can tell that maybe from the clinical data and things like that. So I think that investors are going to look at progress. And today's world, many of these investors are smart enough to know what progress is.
For sure. Yeah. Thank you. Next, we have a few questions on industry trends and future outlook. The first question is, the field of women's health is emerging fast. What do you see as the biggest opportunities for mRNA or other bioengineering approaches in women's health?
Well, I think there are a tremendous number of things in women's health. In fact, I work with a women's health company called Dare. They've licensed actually two of our inventions. One's a vaginal ring, which can deliver really anything. And another is actually a birth control device that the woman could turn on and off whenever she wants and would last for 10 years. And you could put mRNA and those kinds of things to make whatever it is that you might want. But some of that's also going to depend on the biology, but if you have the right drug, say for endometriosis, or you were trying to create, somebody had breast cancer, you could maybe make a better vaccine or a better treatment. There's so many things that you could use messenger RNA for women's health or otherwise.
And I just see another question. With the rapid growth in AI, kind of how do you foresee the possibility of transition from like human generated science to more like AI or super intelligence science? And if you manage to address the problem in computer energy, what would the risk and benefits of such like AI generation be in the science field?
Well, I'm actually pretty positive about AI. I think what it enables you to do is if you have a lot of good data, you can, really make progress much faster. I think the combination of things that you could do in a high throughput screening kind of approach coupled with AI is going to be very powerful. I mean, we've actually used these. People think AI is new. It isn't. It's been around for 80 years. I think ChatGPT certainly showed people what it can do. And of course, computing power has increased tremendously. But geez, back even over 25 years ago, we started a company, Transform Pharmaceuticals, that could use high throughput approaches to increase the solubility of small molecule drugs. 50% of small molecule drugs aren't soluble. And a lot of companies spend billions of dollars trying to make them that way. But what we did is come up with a high throughput approach where we test thousands of excipients. We actually do many at the same time. So before us, people might have done 10 in a year. Now we would do thousands in a couple of days. And then we analyze those structures with AI each time, or machine learning. And then we keep iterating. So each time we increase the solubility. We're also doing that now with a company we started called Vivitex for drug absorption. I mean, there's just so many ways. And when I mentioned the brain on the chip, I mean, that's a way that you could develop, thousands of new targets that you never could have had before and analyze them and use AI to help you figure out, even better targets. So I think it's very, very powerful in terms of what it can do. Of course, obviously, there's some people have concerns over it, but I think it's a very, very powerful tool.
Thank you, Dr. Langer. I had a question on AI for the general audience. What are some of the resources that you can recommend for scientists today to start getting themselves sort of future-proofed on sort of using AI for their research or even for when they're building their own startup? Are there some AI resources and tools that they think you should already incorporate into the day-to-day?
Well, I think there are classes and there are books, there's review articles. So I think there's many, many things that you can use. There's a lot of people that are experts at it. And I think that you can maybe get some of them as consultants and work with you and teach you. I'm sure both at Harvard and MIT. I mean, there are a lot of people that are doing great work. We're actually working with some people at Harvard who are doing great work in AI.
Amazing. We'll explore into that.
I think there's one last question. For someone interested in emerging breakthroughs in the cancer medicine, how would you compare CR ISPR CAS9 versus RNA technologies? And what areas would you advise current postdocs to focus on to address unmet needs in aggressive cancers?
So she's interested in the comparison for CRISPR-Cas9 technology versus mRNA in the emerging breakthrough for cancer medicine.
Cancer, I think, will be mRNA. I think makes probably a lot more sense if you're looking at people are doing like Moderna and other places with mRNA and actually individualized neoantigen therapy, which is training your immune system to attack cancer. And the speed with which you can move to do it, delivery systems are already there. I think so for CRISPR-Cas9, I think it's wonderful. I think that will probably be most useful for rare diseases, certainly in the short run, and for extracorporeal things also. But I think cancer is, the mRNA, I think, is if you're going to try to do in vivo treatments, I would think messenger RNA, at least right now, would be the better, easier approach. And the delivery systems are there to do it for mRNA.
Thank you, Dr. Langer. We are almost at time. We had really good questions from the audience, and it's our honor, Dr. Langer, to have you here with us.